inhibition of metabolic pathways. What is going on with that? We want to specifically target metabolic pathways of the pathogen without targeting metabolic or without interfering with metabolic pathogens of the host. If we can do that, I can get good selective toxicity in this category. Again, metabolism, we talked about cellular respiration. That's not a pathway we want to mess with. That's a pathway in common with bacteria and in humans. That's not what we're talking about. We're talking about alternative, other metabolic pathways that do other things. If the bacteria has it and we don't, then we can get good selective toxicity. We do need to be careful that we don't target any pathway we have in common. So this is another good, be careful. And again, we're looking for pathways we don't have in common. So let's give you some examples for that pathway. Okay, so what is this? Let's say possibilities of metabolic disruption. Again, we have tons and tons of metabolic pathways, electron transport, enzymes, glucose uptakes. We got tons and tons of options. We've got to target something the pathogen has, a pathway the pathogen has that the host doesn't have. So that's what we're dealing with here. All right, let's do an example. And again, there's lots of possibilities. We just need to know the pathways in the pathogen. And then look for the ones that are different. This is one example of a metabolic pathway that we can inhibit, and this has to do with folic acid. All right, first tell me, what do you know about folic acid? Have you heard this word before? What does this mean? I know you're answering me in your head. I hate it that we can't go back and forth, but folic acid, a lot of you have heard about this during pregnancy. Folic acid is in your prenatal vitamins. They talk about making sure you have a lot of folic acid during that. We need folic acid to build proteins, to build amino acids. We need folic acid to build nucleic acids. During pregnancy, you're going to need a lot of it because you're building a whole new life form with a lot of nucleic acids and proteins. So during pregnancy, you need a large amount of folic acid. So that's why we've probably heard about it before. You can get folic acid. We don't make it. Humans do not make it. It's something we need to get in our foods. Spinach has it. I think some different meats, and there's some different foods that you can get folic acid. It's usually included in your prenatal vitamins because our diets don't tend to get us enough. So they want to make sure. The lack of folic acid during pregnancy causes disorders like spina bifida, where the spine doesn't completely close formed right. So if we can prevent that by supplementing folic acid, that makes a big difference. All right, so that's why you've heard of folic acid. That's why we need it. Humans need it, but humans don't make it. All right, however, a lot of bacteria can produce folic acid. So let's talk about the folic acid pathway. We'll come back to this slide, okay? Folic acid pathway. Now, this is not happening in our bodies because we don't make it, but there are microorganisms that can take PABA, which is para-aminobenzoic acid, and take that and convert it. It has an enzyme that converts it into dihydroteroic acid. There's another enzyme in the bacteria that can convert it into dihydrofolic acid, and another enzyme that can convert it into tetrahydrofolic acid, which is what we call folic acid. And that's what's again needed for your nucleic acids, your purines and pyrimidines, and your amino acids. So this is a metabolic pathway. Again, this is not our metabolic pathway, but many microorganisms have the enzymes that can convert PABA into folic acid. All right, each one of these steps has an enzyme, and we know how that works. So this is normal folic acid synthesis. We take PABA, it binds to a particular enzyme, and then converts it into this dihydroteroic acid. So that is our normal pathway in our bacteria. Now, if we add a sulfa drug, this is a type of drug, sulfonamide, that is very similar in structure to PABA. You can kind of see that there's a lot of similarities in structure, but they're not identical. The similarities are in the area where it binds to this enzyme that converts the PABA to that dihydroteroic acid. This sulfa drugs can bind to the active site of this enzyme and serve as a competitive inhibitor I think we talked about this where the drug, the molecule can come into that active site and kind of just stay in that active site. The enzyme can't complete its function, and so it just kind of gets stuck. So this is a drug that's going to stop this portion of the pathway. And if I go back to this portion, this is the enzyme the drug binds to, the sulfonamides binds to this enzyme, and then we keep this from becoming this, which means we've shut down this pathway. We have blocked the ability of this bacteria to make folic acid. We've got a drug that targets this metabolic pathway. The bacteria can't make folic acid, it needs folic acid. We can kill the bacteria. So selective toxicity can be maintained. Now, problem with this is that this is a competitive inhibitor. And if you remember when we talked about that, if I only have a little bit of the drug and I have a lot of the PABA, sometimes the PABA is going to win. Sometimes the PABA is going to be able to bind to the enzyme and continue along this pathway. So this drug, in order to block it sufficiently, we either need to use a lot of this drug or we need to come up with something else, because sometimes PABA is going to win. Now, problem with just increasing the dosage, just adding a whole bunch of these sulfa drugs, is as we increase the dosage, we increase the possibility that we're damaging the host, that we can have some side effects from this drug. And it may be due to this metabolic pathway, but we're complicated and side effects can sometimes happen that we're not even using. So just by upping the dosage can be damaging. And again, taking this drug over and over again can be damaging. So can we do something else to inhibit folic acid synthesis? And I'm going to go back to this pathway and show you the sulfa drug inhibits this enzyme, but there is another drug called trimethoprim. This one we've used in lab, trimethoprim, and it inhibits this enzyme, the enzyme that takes it from dihydrofolic acid to tetrahydrofolic acid. So we can use a drug and shut down this step. All right, so which one's better? Should we use this drug? Should we use this drug? What if we use them in combination? If I use a small dosage of this drug and a small dosage of this drug, the sulfa drug will work. It'll reduce the number of PABAs that can get converted to dihydroteroic acid. It's not going to stop it completely, but it's going to reduce it greatly. So I've got a lot less, and then this drug is going to get anything that goes through. Using these two drugs together, we actually get a better outcome than if we used either one by itself. This is called synergy. When we use two drugs together, sulfonamides and trimethoprim, to inhibit bacteria folic acid synthesis, together they provide a synergistic effect, or it's referred to as synergy. Using both drugs together gives us a better outcome than using either one by itself. It also allows us to use less of each drug. Now again, we are still maintaining selective toxicity because we don't make folic acid. We don't have this pathway. This is not our problem. So selective toxicity is maintained for that. All right, so I'm going to use drugs. I'm going to break it down in two parts. This is just another type of thing. The other thing that I want to point out about these drugs and using these drugs in synergy is that if I have a bacteria that has a mutation, and that mutation allows it to become resistant to these sulfa drugs. So my bacteria is going along, I just have one that has a mutation, the sulfa drug doesn't work on it anymore. That's okay because I'm also using sulfa drugs in synergy with trimethoprim. So if I do have one that develops a mutation and an adaptation and becomes resistant to this drug is going to stop it. And it doesn't get to replicate itself and survive and produce a whole line of drug-resistant bacteria. If I have one particular bacteria that becomes resistant to trimethoprim, that's okay because the sulfa drug will get it. So using these drugs in synergy not only allows you to use less of each drug, but it also prevents the bacteria from becoming easily resistant to these drugs. So using drugs in synergy can help us when we're trying to get drugs that aren't resistant. Okay, I hope that one made sense. Sometimes that gets a little iffy. Bring your questions to Zoom and we'll figure that out together if we need to. I'm going to stop the video now and we'll pick up with nucleic acid synthesis for our next category.